save meiosis for Thursday or Friday. The epididymis, the vas deferens, the gland, seminal vesicles, prostate gland, and bulbourethral gland. And then we'll stop here and say the erectile tissue and ejaculatory pathway for Thursday. So on Thursday you have your first lab quiz, and that will be about 2/3 endocrine. So out of the 6 questions, about probably 3 or 4 questions, if there's 7, a little more, but about 4 questions on endocrine, that's identifying a model as the thyroid gland or the thymus or adrenal gland or the histology that we started on last Thursday, and then a couple questions from male reproductive, right? Because we don't have a ton of the models, we'll look at some histology today, but it will stop here as far as what you're responsible for. So that penile structures and erectile tissue will not be on Thursday's lab quiz. So about 2/3 endocrine, 1/3 male. So 4 and 2, something along that ratio. Okay, and I'll show you some material when we get into actual lab. So in the male reproductive pathway and the female, the gonads develop along up near the diaphragm actually, along the posterior abdominal wall. And in the male, they end up external to the abdominal cavity and the pelvic cavity in the scrotal sac. And anybody have any idea why?
Temperature.
Temperature regulation. Okay, in the male, the sperm continue to undergo the spermatogonia, undergo mitosis, and we have meiotic division. And we'll look at what that means chromosomal reduction-wise. In the female, just the meiotic division. And so the mitotic division is more temperature sensitive, and if the testes were to remain inside the core of the body wall, the male would be infertile. And so there are several functions that help to regulate this particular temperature. Now you have this diagram in your lecture notes on page 46. And so up here at the top is a 5 to 6 week embryo. All right. Now, if you were pregnant, you would be identified as being 7 to 8 weeks pregnant. Yeah, because when you go visit your OBGYN, they ask the last menstrual period, LMP, and the last menstrual period, if you have a normal menstrual cycle, is essentially 14 days or 2 weeks from ovulation. And they don't subtract that, they just talk about, put that on there when they're determining gestation. But if we study an embryo, we're looking at the date of fertilization. Just kind of an aside if you're looking at pictures of embryos and stuff. So in the 5 to 6 week embryo, this is an indistinct identification. By indistinct, you're not going to be able to determine gender. And I'm going to show you a little video, play animation. But this structure right here is an early kidney. Genital urinary in the male, even though we don't use it as much in the female. The genital system and the urinary system are closely aligned, not just because sperm pass through the urinary tract, but the tissues actually induce each other. In other words, chemicals released by developing kidneys affect the development of the gonads in both the male and the female. So along the posterior abdominal wall, an early kidney appears, and this would be kind of an anterior view of the embryo. So this would be up near the diaphragm. And the early kidney, and we'll go over this again when we do urinary system, is identified as the pronephros. It's segmented, so it appears in parts. It's non-functional. It doesn't filter urine. It doesn't make urine. Right? However, it appears to play a role chemically in the induction of the next type of kidney. So that is, as this is disappearing, the next kidney is appearing, and it's a long column known as the mesonephros. kidney, so that's referring to the kidney. So this is what you're seeing right here. This brownish structure, right? And in close association with it, the gonads start to appear. This structure forms, if we were to look at it, and this is part, some of this embryo, I want you to know these terms, but in the overall concept, we're not going to get into real detail. We see this type of a development. This is a cross-section through the embryo, so cutting it through this way. And this is known as the gonadal ridge.
The inner line or the outer?
This bump. Okay, so this. And the developing gonadal cells in the male, what are going to become the spermatocytes, migrate from outside the embryo into the embryo along the blood vessels and reside in this gonadal ridge. So they're going to be developing, appearing in here. And then the mesonephros is back here. So we see the developing gonad. I'm running out of colors. What's going to become the testes here. It would look the same in the female, but of course it would become the ovary.
Mm-hmm.
A tubular structure with connections that grow into the gonad form a duct that in the early embryo is going to be carrying the early urine. And this is known as the mesonephric duct. On the diagram, it's this brownish structure. Okay. As the mesonephros disappears, what we are left behind, I will draw on the other side. And the mesonephric duct remains associated with the gonad and joins with the bladder down here. And this mesonephric duct will now develop into the vas deferens. Epididymis. And structures connecting to the testes known as the efferent ducts or efferent ductules. So as we go through male and female reproductive system, I'm going to talk about structures and what they develop from. So I want you to recognize that the efferent duct, the epididymis, and the vas deferens have their origin as the mesonephric duct. Okay, I'm going to show you a little video that shows that. So I'll just put these over here under mesonephric duct to clarify. So efferent ductual, epididymis, and vas deferens, embryologically, would all be identified as the mesonephric duct. So here you can see the efferent ductules, the epididymis is going to be here, and here is the mesonephric duct. Back to the slide of the adult male reproductive. Let's see, because you may not be familiar with all of these structures. Actually, let me go forward to the testes. Identify those structures for you. So here are the efferent ductules attached to the testes. Here's the epididymis, and here's the vas deferens. Okay. All developing from that mesonephric duct. Now this development requires the expression of a gene present on the Y chromosome, which is why you see the SRY versus no SRY in this image. So if the Y chromosome is present and the SRY gene is expressed, then this duct system, the mesonephric duct system, will develop, and another duct system known as the paramesonephric duct system will degenerate. If it's an XX cell, a female cell, and no Y chromosome, or it could be an XY chromosome, but the SRY gene is not expressed, some genetic disorders. In either case, the mesonephric duct disappears, and the paramesonephric duct will appear. So the person may have testes, but they'll have a uterus and a vagina, because without the enzymes from the SRY gene expression, we maintain the female and we lose the male components. Yes?
On the second figure on the left, is the paramesopher, that one, is that the vas deferens?
No, paramesonephric duct disappears in the male.
Okay.
So it's this one right here. It's still the mesonephric duct becomes the vas deferens. So this is in the female, mesonephric duct disappears and paramesonephric duct becomes
Oh, I see that. Okay. So the vas deferens, can you just?
The vas deferens would be all of, so here would be efferent duct, this would be epididymis, and then all this would be vas deferens.
Okay, I get it. Okay.
Okay.
Mhm.
All right, so let me play this video. If you're looking for it online to play for yourself, it's under your genital development, male, or when we get to female, I'll show you the female one. It's in French, so I'm not playing the sound, but I'll show it a couple of times. Here's the mesonephros, and the paramesonephric duct is in green. You can see that here, Latin, which is the Latin language, so. And this is just showing those filtration mechanisms of the mesonephros that are going to form the efferent ductules. So now the video will put the components back up there. We have the mesonephros, mesonephric duct, this is the bladder. And now the ureters, this is the early metanephric, that's the true kidney. That's the actual functional kidney, is a metanephros. We'll cover that, that's urinary. But there's no other organ embryologically that has three different separate, we don't have one heart and then another heart and then another heart. There's an ongoing. In the kidney we have three separate. So here's the gonad, here's the testes. Now the mesonephros is degenerating. The duct system, these would be the efferent ductules associated with the testes, and epididymis, and vas deferens coming down to join into the urethra. Okay. We'll look at it again. The blue is the degenerating female duct, the paramesonephros, which in the female, the reverse would happen. Okay, so let's play that again. I was talking when those gonads showed up. So mesonephros and mesonephric duct. Filtration structures of the first functioning kidney. And then we'll put the mesonephros back on it. Don't worry about the kidney development, we'll pick that up with a different system, but again, it's an entirely different embryonic structure. Now you can see the first testes appearing up there, and here's the paramesonephric, the blue, developing. It's going to appear and then disappear. Notice that the gonad gonads are moving down the posterior abdominal wall. They're gonna end up even lower than that.
How they start up there and
The reabsorption of the